Okay, so let's get started. Kia ora koutou. My name is Jill Dobby, and I'm a machine learning researcher and a professor in computer science here at the University of Auckland. It's my great pleasure to welcome you to the 2022 Ihaka Lecture Series, which is sponsored by the New Zealand Statistical Association. As you all probably know, the Ihaka Lecture Series takes its name from Ross Ihaka, who along with Robert Gentleman created the statistical programming language here at the University of Auckland in 1993. It's amazing the influence that the language has had worldwide, and in 2021 it was ranked 11th in an influential index that ranks the world's most popular programming languages. Of course, a large part of its success can be attributed to R being under a free software license and subsequently the large community that has grown around it. I continue to be amazed by the incredible people that are around me, and Ross is no exception to that. It's hard to overestimate the importance of his contribution to both statistics and computer science around the world. The Ihaka Lecture Series has covered a number of topics over the years, ranging from topics such as the risks and benefits of sharing personal data to the interface between statistics and computer science. This year's series features 3 speakers who develop software tools for data science. So the title of the series this year is Building Building Blocks for Data Science. The subsequent lectures in this series are next Thursday and the following Thursday, so that's the 4th and the 11th of August. So again, welcome, and I hope you enjoy the 2022 Ihaka Lecture Series. I'll now pass over to Beatrice, who's going to introduce the speaker tonight.
All right, so it's my pleasure to welcome Emmy Tanaka, who comes to us from the Department of Econometrics and Business Statistics at Monash University. So she has had an interesting trajectory through different areas of statistics and computing and through different institutions in Australia, beginning with her PhD at the University of Sydney with a topic in statistical bioinformatics, with most of the computing done in Python and Bash. She then moved to University of Wollongong, and then back to the University of Sydney, and finally to Monash in 2020. So along the way, she's added interest in software development, statistical workflows, and data visualization. And tonight, she's going to show us how those things combine with her interest in the design of experiments to create the grammar of experimental design.
So can everyone hear me? Okay, kia ora everyone. Thank you for having me today for the Ihaka lecture. I'm very honored to be here to talk to you about the genesis, or essentially the start of experimentation. So you are probably aware of the recent global pandemic, as I can see many of you are wearing the mask. And if anything, these events is really a reminder of how important science is. And in particular, the development of the COVID-19 vaccine wouldn't have been possible without these experimental trials that can give confidence to the actual effectiveness and the safety of these vaccines. So it's not just about the global pandemic, right? It really, the decisions or results that come out of experiments really can affect everyday life. And for example, like what diet is effective in lowering your insulin level? Is lookout laws effective in lowering alcohol violence? Effectively meaning like, does policy actually work? So what brand of washing powder is most effective? How much fertilizer do we want to use for plant growth? So there is a large aspect of our lives that are improved from the knowledge that we acquire from analyzing experimental data. So how does an experiment begin in the first place? Well, in general, you have this originator of this experiment, who is what we call the domain expert, who drives the experimental objective and has intricate knowledge about the subject area. Then followed by, we have the so-called designer of the experiment, which will be typically an individual that's labeled perhaps as a statistician. And the statistician will take into account the statistical and practical constraints in order to generate the experimental design layout. Followed by, we have the so-called executor of the experiment. And I'm going to call this person as technician. And this technician essentially carries out the experiment as per the design that's been supplied, but also collects the experimental data as well. But lastly, we have the digester of the experiment. And this is essentially, you can think of it as a person who analyzes the experimental data. And it may be the same person as a statistician, but also it may not be. So all these actors are really purely illustrative. In practice, we could have multiple people taking on each of the different roles. And also, we can also have one person taking on multiple roles. A person who takes on a particular role doesn't necessarily have to be specialized in that role. And for example, experimental design doesn't necessarily have to be generated by somebody that's really thoroughly statistically trained. It could be generated by the domain expert who may not have had the thorough statistical training in the area. So the roles are rather fuzzy. And even if we have a very clear set of roles, what happens is that these different individuals will have to still communicate in order to share what they know to really conduct an experiment. So the thing is, communication is inherently fuzzy. And this is where we can kind of learn more by looking at the area of communication theory. So one proposed communication model is this transactional communication model, where I've just depicted here, where we've got two people. Here, one is domain expert, the other one is a statistician. And essentially what happens is that as a domain expert is passing along their message, there's this noise that comes in place. So by the time the message gets through to the statistician and the person has to decode the message, that decoded message may not have been exactly as intended by given as the domain expert. Now, that's one transaction, but then of course in the transactional communication model, what happens is then there's a response coming back from the statistician, and then again the domain expert will respond back to that communication and so forth. So what you will see in practice is this a back and forth communication between the two individuals, and along each time there is a level of noise that gets encoded in there, and some of them might be less, but nonetheless we have to acknowledge that the communication is indeed fuzzy, and so this transfer of knowledge in order to actually properly do the design of experiment is actually hindered by this fuzziness in communication. So let's look at an example. Let's say that in this comparative experiment, we have some treatments of interest. In this case, we're looking at whether a diet works for lowering the insulin level. And I've got two levels of treatment. One is a control diet, and then the other one is a high sugar diet. So the domain expert will explain and say, okay, we're going to be measuring the insulin after 4 weeks, giving the treatment to the subjects. And then we will make conclusion from this. And then as a statistician who is going to design the experiment, you will think about taking into account some of the experimental design principles, including the concept of randomization. So the randomization process means that you're going to basically, for the given number of subjects, you're going to randomly assign them either of these two groups. But, in practice, what happens is that experiments can be modified during that conversation. So here the domain expert might have said, okay, actually, we want to include the high-fat diet as well in terms of comparison. So you put that in, but then also there's possibly some experimental structure in that experiment, right? Which you have to put in your designing process in order to do your appropriate randomization or optimization process. So this, again, you require that information from the domain expert. And in this case, we're looking at the diet for effect. So essentially, there might be like a certain grouping that occurs within your units. If this is a human experiment, you might think that, and then knowing that diets, the effect of a diet on the insulin level might be biologically driven, you might want to look at the sex of the different individuals and stratify your randomization according to different sex. Age is also a factor, as well known in terms of the insulin. So again, you might want to also block things according to their age as well. So this is something that you really, in order to do your experiment, you need to extract that information out from the domain expert. And my proposition is that communication is really the key to get the most out of the experimental data. Without really getting that information, you can't really do a design well. So while we like to think that, coming up with a complex design is good in getting out the most efficient information out of it, really without having extracted the relevant key information, you won't be able to do the design process well enough. So I want to talk to you about another aspect of the design, and that's the design of the domain-specific language. Now, for those people who don't know what a domain-specific language is, that's a computer language specialized in a particular application domain. And R is one of the most popular, as you just heard in the introduction, DSLs for statistics. So if we look at experimental design R packages, well, first of all, its packages itself is internal DSL. And there are over like 100 R packages for the design and analysis of experiments. And in contrast, if you look at other languages, there's like only a handful. And as an example, like a Python only has a handful of packages. And if you also look at the output of the designs from Python, the range is also quite limited as well. And I bring this up as an example, not because I'm trying to make it a battle between like the languages and saying one of the languages is better. It just tells you that really R is like a place where I think a lot of statisticians contributed, and particularly experimental design contribution is quite large in that area. So if you look at this set of packages, you can find actually that a lot of the popular ones are very using the recipe-based functions. And I'll show you in a minute what I mean by this. If you want to know more information about this, I have a paper on this that actually talks about the more thorough analysis of these experimental design R packages. So let me start with showing one of the most popular ones. So this is the Ugly Collab R package, and this was the top downloaded packages like for many years. And so if you look at this, so in the first instance, what I'm doing is just describing like a vector of the treatments, so the different diets. So I have 3 levels here. The very first design that I'm generating using the Agricola package is the completely randomized design. So if you look at the name of the function, it's design.crd. And the CRD is essentially the acronym of the complete design, randomized design. So if you don't know what this is, essentially what happens is that you just have a set of subjects, set of units. And in this case, we got a replicate of 10 for every diet. So there will be 30 altogether in terms of the subjects. And then here we're passing in the diets as a treatment, right? And then the complete randomization means that there's no structure on the units, nor is there any structure on the treatments as well. Then we have the randomized complete block design. In this instance, what happens is that there are 10 blocks, which are each equal to 1 replicate. And within each block, what happens is that every treatment only appears exactly once, and it's randomized within each of those blocks. So if you look at the name of the function, again, here it's design.rcbd. And then that function essentially is an acronym of that randomized complete block design. Then let's say that we have another treatment factor. This time we're going to include exercise, which has 2 levels, whether that subject had an exercise or not. And we can generate a factorial design. This one less obvious in terms of the name, but it's design.ab. And then what happens is that you pass in the number of levels for each of the different treatments factors. And again, we have a replicate of 10 here. Split plot design. So this is like a special design. So this particular named design means that you've got a factorial design with two treatment factors. So you feed in each one of them. So treatment 1 is going to be the diet treatment. Treatment 2 is going to be the exercise. And then we'll have 10 replicate. Now, that's the structure on the treatment, but there is a structure also on the units. And the way that the units work is that there's a nesting structure. So essentially, the small units are nested within the big units. And also, the allocation is also special as well. Where instead of completely randomly allocating the treatments to the units, what happens is that one of the treatment factors is applied to the big units, and then the other treatment factor is applied onto the small units. Now, let me show you the other one. This is ARG design. And this is a class of, it generates a class of optimal designs where a particular model is given. And based on the user supplied, chosen criterion, it's going to algorithmically find the permutational treatments such that it optimizes the criterion. So in the first instance, what I'm doing is to create like a candidate list of the treatments. And then here, what happens is that the function, the opt_federal from ARG design, is going to create an optimal design according to the federal's exchange algorithm. So first instance of the argument is essentially the model that you specify. And then you feed in your candidate list of treatments, number of trials, and then also like a certain criterion. In this case, I've picked the D criterion. Another function is using optimal design via Monte Carlo approach. So very much similar, but the algorithm on the hood is just different. And I think this is actually not too bad in a sense that it is quite general. And unlike the previous one where we saw the recipe-based design in terms of the randomized design, this has a little bit less constraint of like what you need to feed in terms of the structure you get. But you may have noticed in both packages, you really can't infer the context quite well. In particular, did you realize what the units were in this particular treatment experiment? So reading the code, you can see what the treatments are, but you really don't have any information about the units written out. And while you might have imagined that this diet experiment is on humans, the actual base of this was based on animal study. And then now that you are given that context, as a designer of the experiment, you may have certain domain knowledge, or you may have acquired domain knowledge from your domain expert collaborator, who would have told you that, look, pigs come in litters. And so when we're doing this experiment, a natural block would to really block it according to litters as well. So that's actually a unit structure that you would need to put in consideration when you want to create your design. So in part, we have, in just looking at the code, certain informations are actually not written. And you may normally, when you start doing the designing process, you may have had this implicit understanding. And so you may have put the blocking structure appropriately within that. But say another person will come, just like later on, we will have an analyst that comes later on, which could be a totally different person. And this is very likely because of the fact that time elapses between experiments. So that person comes about new and looks at your experiment design code, will not be able to tell what exactly happened. So certain information may get lost along the way because a lot of the information were implicitly known to people. And so, but then they weren't ever explicitly written somewhere. And then the knowledge of how the data was generated actually can also affect how you're going to do your analysis later on. So let me talk about the work that I've been doing in this particular area. And I've been kind of like writing up a computational system about this. And I've been putting it in a book called The Grammar of Experimental Designs. And it's not complete, but if you want to see like the progress, you can always go to the link, emmytanaka.org/edible-book. So much of this work is really about this abstract and concrete syntax. And this is a concept coming more from computer science. So the definition of a concrete syntax is that it's a reference to the external representation of a language. And when I say abstract syntax, that refers to the internal representation of a language. And what I'm doing in this the grammar of experimental designs is essentially proposing a design of these abstract and concrete syntax. And essentially, I'm putting forward that by doing this, we can aid the intrapersonal and interpersonal communication that's ultimately going to help you design the experiment. So to be more specific, when I say intrapersonal, so this is actually the code that you see that you've written up. The way that it's been designed, the syntax itself, I think can help users clearly express as well as abstract and clarify what the desired experimental design is. So intrapersonal is just like your communication with yourself. And also, now that you've explicitly written it out, the communication is no longer just by yourself as well. It can now be read by other people. And this is where the interpersonal part comes in, where other people can read your code and have an understanding of what you understood at the time and the intention of that particular code, like a design as well. Okay, so it's best to see an example. So I've got this example, which comes from a paper in ecology. It's looking at the plant diversity effects on soil microbial functions and enzymes on a glass lung experiment. So they had some hypotheses. So one is increasing plant diversity leads to increasing soil microbial biomass and enzyme activity. Higher temperatures decreases soil microbial biomass. and enzyme activity. And the third hypothesis was higher plant diversity buffers effects of elevated temperature on soil microbial biomass and enzyme activity. So you can have a look at this paper, but I'm just going to strip down to the experimental design part. And this is like the full description of it, which is rather wordy. And because we won't have time to carefully read through this, I'm just going to present like a condensed version of it. So I'd like you to have a read of this. And then while you are reading this, have a think of like, what is this experiment structure? Like what sort of units do we have? What's the treatment? What's the location? So I'll just give you a few minutes to have a look first. Would anyone like to volunteer, like what you think the experimental structure involves or experimental design involves?
No takers.
No takers? I know some people who are very good at experimental design, so I'm pretty sure some are in Auckland, so I'm pretty sure some of you already figured it out.
Clarification, the environment when you're taking the experiment, is it internal to like?
So I heard some, but
Internal planting like in a hothouse, or are we dealing with an external plant like an agri farm? Like you
so
plant a
this
market
one is
garden.
actually, this is outside. So it's not inside like a glass house.
Okay, it's just that you mentioned there about the heat treatments emitting 600 watts. If
Yes.
that's what I read right, and that way you've got variance of temperature control of the environment can affect the soil and the growth rates of your plants. Whereas in a hothouse, you have got temperature control that you can vary depend on the external factors, that if it's raining outside, the hothouse will have certain environmental conditions that will change.
Yeah, so they do show the setup in the actual paper, and they have a picture. It looks like it's set up outside, and they have some kind of thing that I think radiates heat from it. Do you kind of know what's the treatment factors in this particular experiment?
The heat and the number of plant species.
Yes. So we're looking at the diversity, and I'll just start highlighting. So plant diversity, and we're looking at the gradient of 1, 4, or 16. This is actually randomly chosen from the set of like 19 species they have. So that's one of the treatment factors they're looking at. Unit-wise, there's this part that says plots were divided into 3 subplots. So this is kind of describing a bit of hierarchical structure within the units. So one subplots are going to be nested within the plots. And then we got the heat treatments. So this one specifically says it's applied to subplots. And then if you look at the different heat treatments, we're looking at so the whole thing about the watts is about how they applied in order to get a certain constant increase in temperature. Whether that's actually true or not, I do not know. I have to assume that's the truth given what's been written. But this causes essentially an increase in 1.5 degrees Celsius increase. Another one is 3 degrees Celsius increase. And there's the one that's controlled. So there's no heat treatment that's been applied into that particular plot. Now, there's 27 experimental plots in this one. And so, in fact, if you look carefully at this is actually a split plot design. So you can see heat plots applied to subplots, and plant diversity applied to the bigger, like the plot itself, the experimental plot. So let me show you how the code will be using the system I have built called Edible. So Edible, once you load it up, the first instance is that you're just going to initiate the design. So the design here, I'm just passing in the name of the particular author of the paper, and that can be anything. Next instance, I'm defining the units. Here, I'm specifically saying that I've got two sets of units, the plot and subplot. Plot, there are 27 levels. And in terms of the subplot, well, we've got it nested within the plot. And then for each plot, we've got 3 subplots. Then the treatments, we've got the number of species. So 1 for 16, which is measuring the diversity. And then we've got the temperature, right? The 3 different temperature levels. In terms of what treatment gets applied to what unit, well, the diversity, number of species is applied onto plot. Temperature is applied onto subplot. Then we're going to assign. And how are we going to assign it? This is random. And this is a part where you can modify things to other kind of assignment. And arguably, I would say that this is actually the component where a lot of people focus on when you are doing the designing of experiment, how to appropriately do the assignment of the treatment onto the units, given the constraints that you previously defined. Here, what happens is that I've got a very simple kind of application in there. But of course, what happens is that this part can be substituted by any other functions instead. Then you serve table, and serve table here is an indication, basically saying that, okay, I've defined all my components of the experiment, and I'm now ready to get the experimental design table. So that's what you see at the very end. So you get produce an edible, which is the name of the package, stands for experimental design table. Or if you are a tidyverse user, it's experimental design table. So you can get a data frame back. And essentially, this actually has the allocation of the treatment factors to a particular unit. So why edible? So the way that I saw with this is that the semantics of the syntactic sugar are really aligned with basic terms in experimental design. Before, when we had a look at the agricole package, when we had that recipe design, we don't really get reminded of what the units are or the treatments or even what the allocation process is. But here, instead of actually using a recipe design, what happens is that I'm actually thinking about it more in basic terms. Also, the way that the code's written can be used for communication to capture the user intention. Experimental structure is built step by step. So you've got the abstract syntax that allows for incomplete experimental structure. And so this is part of the innovation, I thought, is that a lot of times when you try to create an experimental design and you face the software, it seems like you can't have like a incomplete understanding of the current structure. Like you have to be able to have like a complete set. You need to know exactly how much replicates you're going to have, what set of treatments you have, and so forth. So the part about this in the edible is that it's kind of delineated. So you can do it step by step. If you know what the treatments are, you can start writing it out. If you know what the units are, you can write out that in another step. So there's a part where like the whole object is stored, if you don't know the full structure, as an intermediate product, which is on the core of it is just a graph. Then the concrete syntax is being made easy to replace because now everything is like a single step. And so what happens is that, like, if you find particularly that you don't like the way that I assign the treatments onto the units, which you rightfully probably shouldn't, because it's hard to actually make a very general assignment without actually making sure that you get like an assignment that's specifically catered for your situation. So you might want to replace that step. But then instead of having to do all the other things, it's just basically one single step that you want to modify rather than redoing the whole thing from scratch. And the last part is that there's a lot of downstream benefits. And this is essentially one of the things that I thought it's very important to have because it is actually a lot of work in order for people to write it out as you compare to the syntax that you saw before. And so there needs to be, something in it if you're going to commit something like that, besides from the principal point of view that you're actually clarifying the different components. So some of the downstream benefits can include things like easy and quick visualization. So this is an extension package I've built called dgust. Now, this one is still a developmental and it's going to probably rapidly change, but I want to just present the idea behind it. So dgust is essentially, you can think of it as design of experiments as ggplot object. So DEGG, and also it's short for degustation, which if you go out in restaurants, you get a degustation menu. It's essentially an appreciation of your food, right? And so this is a case where when we're looking at visualization of your experimental design, essentially it's an appreciation of your experimental design. And really the idea behind this package is just a simple function called autoplot. And what you do is you pass in the edible design object, the final design, into the autoplot. And you've done this hard work of defining what the unit structure as well as the treatment structure and its allocation are already, right? So that information is encoded within the object. So the idea behind this is that now what you want to do is to leverage that information in order to quickly make like what might be a good plot. And the quick part is important because then you can do this quickly, perhaps when you are talking to your domain expert collaborator, and just quickly clarify whether this is the correct structure or not. So you can see the output you get on the right-hand side. And so you see here that there are 27 experimental plots, and there are 3 subplots. So the circles that you see are the subplots, and there's like 2 circles. One, the outer color actually shows you the diversity, so the number of species, and color map to the number of species level. The inner circle shows you the temperature treatment, and then the colors basically map to that particular treatment factor. So you can see how it's been allocated for the particular different experimental plots and subplots. So quick communication is indeed useful, but because it is a ggplot object, you can actually customize this visualization using your existing knowledge if you are already a ggplot user. So what's happening here is that I'm just modifying what the design I had before. And then, you might want to actually publish your experiment design. So you want to polish it up. In terms of how you modify it, well, it is a ggplot object. So you can just use whatever ggplot functions on top of it, to customize it. So anything you see from line 6 to 17 is essentially not from my package, but from ggplot package. And you'll see the modification that you see on the right. You also have actually ability to substitute the units so that it's not circle. So if this is a case, it's a plant study, you might want to use a picture of a plant, and then you can pass in a picture of a plant and get like a product out of that. So at the moment, it's a bit slow to do that. So there's a lot of work that I need to do in order to polish this up. But this is a general idea that in part, it's to enhance communication. Visualization is indeed one of the parts that can be helpful. And if you can get a quick visualization, great for communicating on the fly with your collaborator. If you can get like a polished up visualization, that might be helpful for the technician as well, right? To get that plan to follow. And you can also use it for publication purposes. Now, there's another downstream benefit, which is to set record. And by record, in the edible system, this refers to variables that you intend to measure or observe. And that's generally talking about the responses. So a lot of time when you do design, you don't really need to know the response itself, but actually explicitly making a statement about it can be helpful. And so you can do that in edible. And on top of setting the records, you can set expectation of those records, right? So write down the plausible value of those records. You can also do simulations of those records. And that's why I quickly simulated and do mock kind of pseudo analysis on it, see how it is. And that simulation actually can take the information based on what you've set in your expectation. You can also export your data, which contains data validation. And so this is a case where if you have set your expectations, and essentially if your record is something like looking at the biomass, then a biomass can only be a positive number. And therefore, any kind of input that goes into the data should always be a positive number. So when you export this, then if a technician tries to enter a data that is actually not within a valid range, so a negative number, character, so forth, it actually wouldn't allow you to input that. So I'll show you the syntax for this. So here we've got the design from before, and we pipe it in so that it goes to the set records. So the left-hand side is a new variable that you're creating called the microbial biomass, and you're measuring it onto the subplot. So the right-hand side is an existing variable within your design. That just is quite boring because all it does is create a new column, and then it has like no information on it. It just tells you that it's a record. And at this point, it's just signaling your intention to capture like what you're going to measure. The next step is this part where it's setting the expectation that this response is going to be a positive number. So I'll just show you like a live code of this, so you can actually see how it works. So I've got that, and I'll just show you one line at a time so you can see. It just does nothing like when initially initiating it, and then you can go on to do step by step. The printout just shows you like what the graph structure may look like. Internally, you can't see a data frame as yet because you haven't signaled it. And even if you signal it, let's say I say something like this. So you've got plot equals to 27, subplot equals to 3. I haven't defined any relationship between the two of the units. And then you try to get the table from this. What happens is that it doesn't output anything because it can't reconcile to understand how these variables should be related to each other. So the table only shows where if if the different variables can be mapped to a single observational unit, and then you will get like a valid output. So all this is just basically the temporary structure. and then just holds as a graph information until you get to the final part. Wait, we've got that table. And go to the record part. So the record, again, quite boring, just creates that extra variable, nothing written there information. This one also doesn't really tell you in terms of much because it's kind of the validation, the data validation is just encoded behind the scenes. But if you save this, and let's say that I'm going to export this design now. The file name, put it in the downloads folder. My design.xls. Now it's been written out. So let me open that for you to see. And so here's the file. And then if I go to the data column, I've got this part. Now, if I try to like enter like a negative number, it's been encoded. So that validation rule is already in there. So that's what I mean by the expectation of the record and how the data validation comes in play. In terms of the simulation, this is again like work in progress. But what happens is that you can simulate records and say that microbiome biomass is going to be normally distributed, and then give it the mean and standard deviation associated with it. And when you do this, what happens is that it will generate according to that. And some NAs are created in here because I've chosen the values so that it's more likely to get negative numbers. And this has been censored by default to be like missing value. But you can censor it to be positive value, like 0 or other kind of values as well. So, and this part here can also be based on like the terms in the other one. So you can actually do it according to the number of species or temperature and assign different effects for it. So it's a bit more realistic than assuming that it's, the whole thing is generated from a normal distribution. So the main thing about it is that the data is encoded as an object. And this allows you to actually use this information. And so some of the things you may not really agree with, and that's okay. But the main thing is that it's been encoded. You don't like particular functions that I've created. Please feel free to let me know if so first. But also you can easily replace it as well. You can just use the encoded information in the way that you like instead. Now, another thing you can do is to do unbalanced design as well. And the syntactic sugar for this, I think, has made it a little bit easier than the way that maybe you typically would do it in terms of like a procedural kind of programming style. So in this particular experiment, we actually had this plant diversity with 1, 4, or 16 species. And these species essentially were taken from like a random choice of 19 species, right? So I could kind of create like a similar scenario based on this. So 27 plots, and such that the first 9 is going to have 1 species, the next 9 is going to have 4 species, and the next 9 is going to have 16 species. So here, like I don't have to have like a exactly same number of units within a particular larger unit. I'm allowed to get like a different values. And for those people who are familiar with tidyverse and use case when, the syntax is very similar to that essentially. After I do that, it's essentially like treatments here can be thought about with the 19 species, and you're assigning the species onto the plant, and it's a random assignment. And we can have a look at the quick visualization to see indeed this is actually done the correct structure. So if I output this into it, you can see that there is indeed 1 plant only for the first 9, 4 for the next 9, and then 16 for the next one. Now here, what happens is that there's going to be too many colors. If there were 19, so I've just like got an option to say that the max is going to be 3, and everything else is collapsed into like other category instead. Okay. So now you might realize with this work that the whole designing of the abstract and concrete syntax is somewhat subjective. And there are some opinionated choices, but what I found kind of helpful is to read work more in the computer science. So if you do this kind of thing as well, I find it very helpful to look at more design guidelines for domain-specific languages. And definitely a lot of people, I think, in computer science have already thought about this. So I think I try to think of it in their terms and try to leverage what they've done. Some of the concepts that come up quite often are, for example, consistency, using and leveraging other work, and then trying to have it so that there's a bit of a constraint in number of elements that you make. So don't keep on adding more functions, like have it so that it's a bit more concise. So while you want to allow users to be able to express, but also by having a certain constraint in the way that they can express a certain concept, makes it easier to standardize things, and also use essentially language that already a lot of people will be familiar with, and you want to try to enable modular kind of functionalities. Now, so today we are at the Ihaka Lecture, and I wanted to also acknowledge the great work that Professor Ross Ihaka has made as the original creator of R. This work I really present today really builds on the shoulders of many giants, and without having leveraged all the contributions that are made by the R core team, Tidyverse team, and the wider R community, I wouldn't have been able to get this far. So thank you again for having me today and listening to my talk. Just to summarize, so experiments are human endeavors, and we have to acknowledge that there are inherent flaws to the way that as humans we communicate and can understand with each other. So I think even among this fuzziness though, if we have a framework, it can shed more clarity around it. And if anything, I think that's the way to really tackle a good way of getting the most out of experimental data, keeping in mind the garbage in and garbage out principle. We might like to think that sometimes having really complex analysis is great, but then no matter how good your models are, it really won't save you by doing that complex analysis if your data itself is garbage to begin with. So you can find my slides at emmytanaka.org/slides/Ihaka 2022. And also, if you like to play around with the packages, Edible is on CRAN. And if you want to play around with dgast, at the moment, it's only available on GitHub. You can also find the book that's in work in progress and other things. And if you have any kind of feedback or comments, I'd very much love to receive them. So feel free to contact me as well. Thank you, everyone.
So we have time for questions, and we've got some lovely helpers that if you have a question, they will bring the microphone to you so that Emmy can hear your question.
My question is, what template did you use for your R Studio?
Oh, what template?
Yeah, to get
like
oh,
that one. I think it was the midnight. The tomorrow midnight bright.
All right, cool. Thank you.
Hi,
Thomas?
Amy. Can you get sort of printed layouts for things like, for designs, for things like 96 well plates or whatever, out of the software. It seems like that would be something that the plots would also be useful for. Would be getting pictures of layouts for
The picture of the layouts.
Yeah, so for putting on a bit of
like in the export.
or in a export, or in a, if you were in a lab study, if you wanted to say which things go in which spots on a 96 well plate or something like that. Because these are pictures of the design, but they're not pictures of the experimental material.
No, they are not. So I guess that would be like a substitution of the legend. At the moment, so this shape, I think it stays the same as a circle. But you're right that it probably would be nice to actually have this as like the actual experimental treatment. Is that what you mean, Thomas?
I think it's more like you've got plot 1 to plot 27 there, and some like keen plant people might want to know like what the layout is north, south, and that kind of thing. So
yeah.
And I mean, I think it's the same thing with the plate.
Yeah, exactly. The same sort of thing with the lab.
Yeah, so in that sense, then the user needs to specify like the coordinates. So you could do, in particular, like a row column structure. So there is a function within here that actually does allow the that sort of like a two-dimensional design. And if that's the case, then the output is going to be like it will be like a layout as you expect in a field. So let me just show you an example. I don't have it on my, but if I go to cookbook, and then let's look at the Latin square design. So got row, column, and then the unit is essentially across of the row and column. And so what you would do is that if you have this structure, you will need to define this. And then the autoplot for this one is that it recognizes that it's this row column design, and then it will choose based on that. But without this information encoded, you won't be able to infer it automatically.
Kathy?
So, nice talk, thank you. Paul, like a few years ago, I worked on an experimental design, and in that design, we were working with rat livers, and rat livers are different to human livers. They have these lobes, and so what they were doing was every hour, or maybe it was less, every half hour, they would cut a slice off one of the different lobes because they were believed to have different functions. So the scientist asked me if I could do a diagram which showed the random order in which the lobes would be sliced. And actually, Paul, he doesn't remember, but it's about 5-6 years ago, and he helped me. So in that case, he had a book which said on this day, this rat is going to have lobe 3 sliced in hour 1, lobe 1 sliced in hour 2, and so forth. So that's what he had. So is that your sort of idea, Thomas, where you've got an image that's really quite specific to the experiment? So you can do that sort of thing. So that's just a long comment, sorry. But my other question is something that I'm interested in, and that is that you have the data validation built in, which is kind of really nice. And I was wondering what you use to create that when you export, that you actually have that the validation rule is being exported to Excel. What did you use to do that?
Yeah, so this is based on a package called OpenXLSX. And essentially what happens is that the data validation is encoded via the Excel route. I can't do a CSV in this instance. So if you look up that package, it's really coming from that.
Oh, that's great. Thank you very much.
Charlotte?
Kia ora, Emmy. My name's Charlotte. Great talk, thank you. I think hopefully a really simple question, coming from the idea of teaching this kind of stuff to biology students. I love the idea of the step-by-step piping, which I guess is a tidyverse thing. Is there scope to then basically have a final line after that simulate data that you showed us to, I want to say analyze data, but you know what I mean.
I know what you mean. So that's a model. Yeah, I actually thought about that. And then I was also very careful, because that means that people might think that model is like the absolute one. So if you go to the issues, and if you have comments about this, I would love to get your ideas of what the syntactic should be. So like I was thinking like suggest analysis, suggest model. I mean, I gave a talk and somebody else said design model, baseline model, and so forth. So the information is indeed there. If I go this one, oh no, I think I have to go. So there is sort of like information encoded, and there is a part of anatomy. Yeah. So it includes the unit structure. Separately, there's a treatment structure that's embedded. So there's like a little model in there already. But then I haven't implemented anything about like suggesting models yet. Particularly, I want to be very careful doing this, just in case that's going to encourage people to think that the baseline model is like the only model that they need to use for the analysis. So if you have ideas of like what you might think is best practice, I would very much love to listen.
Brilliant. Probably avoid it and let people make their own decisions.